Hello, 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 Science Savages. I'm Nick. I'm new here. I'm joining the Science Savages today to teach you about mitosis. Boom. Mitosis comes right after interphase in the cell cycle, and has a total of 4 stages. You got prophase up first, metaphase second, anaphase third, and telophase is last. First thing we're going to talk about is prophase. Summed up, prophase takes this thing called chromatin, DNA in the nucleus, and condenses it into this other thing called chromosomes. Chromosomes are made up of 2 stretched out V-looking things called sister chromatids. Each of these things holds identical copies of DNA. In the center, there's this thing called a centromere that basically just holds the 2 of them together. Chromosome is now a singular structure that holds the DNA replicated during interphase. Farther into prophase, the nucleolus disappears without a trace, along with the nuclear envelope. Spindle fibers form and move to opposite poles of the cell. These little guys then stick to the sister chromatids in opposite poles. This makes it possible for each cell to receive a complete copy of the DNA. The next phase is metaphase. Metaphase, you can remember it by thinking of middle phase. During this, these sister chromatids are, moved to the middle of the cell and into a, line formation. I mean, that's pretty much it. It's one of the shorter steps of mitosis, but it is extremely important because without it, then it wouldn't allow the new cells to have accurate copies of the chromosomes/DNA. So without it, it would not end well. After metaphase is anaphase. In anaphase, the chromatids that were sitting in the line are pulled apart by the spindle fibers that have been attached to them. Pretty much, it's like it just gets taken opposite ways, and it looks like, A's going in both directions. When separated, the chromatids move towards the poles of the cell because the spindle fibers are pulling them there. The last phase of mitosis is telophase. The chromosomes group up at the poles of the cell and decondense. Pretty much, like clown cars. There's like 10 of them, and then clowns pretty much just seem to be multiplying because there's just so many of them. Yeah, no. The decondensing causes 2 brand new nuclear membranes to start forming and the nucleoli to start unvanishing. This is pretty much the end of mitosis. Just a little side note. Out of mitosis comes 2 new cells that are known as sister cells and are identical to the parent cell. So, you got like parent cell just up here, then you got like mitosis, and then it's like boom, you got 2 sister cells like over here. So yeah, thanks. You're welcome. And now on to Levi, teach you a little thing about meiosis.
Hey guys, as Nick already said, I'm going to be talking to you guys about meiosis. Meiosis only creates sex or gamete cells. In the human body, there are only 2 kinds of gamete cells. The one in the woman is called the egg, while the man's is called the sperm. Meiosis only occurs in eukaryotic cells. There are 8 steps, not including interphase and cytokinesis. The 8 steps are prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 2, metaphase 2, anaphase 2, and telophase 2. The first 4, prophase, metaphase, anaphase, and telophase 1, are all very similar to what Nick talked about, the only difference being crossing over. Crossing over occurs between prophase 1 and metaphase 1. Crossing over is when chromosomes switch with different parts of the genetic material with other chromosomes. This causes for genetic variation, and it's how we all look different from our parents and our siblings. Genetic variation is good because it means that one sickness can't wipe humanity off the face of the earth, which is really nice when you think about it. So we go to prophase 2. Remember that 2 cells have been formed already because of the first part of mitosis. They're not sister cells because they have different DNA. And like prophase 1, the membrane disappears and the DNA turns into chromosomes again. Then we go to metaphase 2, where the chromosomes line up in the middle. It's not much different from metaphase 1. Then we go to anaphase. Now what makes anaphase 2 so special is that we only have the number of chromosomes for one cell in our cell. So when the spindle fibers take half, there's only half of the chromosomes remaining. This is good so that when a male gamete cell meets the female gamete cell, we don't have double the number of chromosomes. Lastly, telophase 2 is like telophase 1. The cytoplasm starts to pinch in the middle and cells divide. That's all for me today. Thanks for watching.
Thank you guys so much for watching. Don't forget to smash, decimate, and destroy that like button, and subscribe for more daily science videos or whenever we get back on the channel. Thank you. Goodbye.